All right. Thank you, everybody. I'm happy to kick off this meeting, and I appreciate invitation. My name is Michael Tsipursky. I'm the CEO of Biotechnology. We are developing oxygen nanobubbles, nanoparticles, for the treatment of hypoxic conditions. We're first-in-class nanotechnology allowing targeted oxygenation to minimize tissue damage resulting from hypoxia. We are addressing serious unmet need, and we have a unique solution showing efficacy and various advantages. We have a swift regulatory pathway, and it is a platform technology. We have a strong team, which was founded by myself. I'm a physician, a retinal surgeon, and a professor, Iraduaj, who is a renowned nanotechnologist. We have a strong support team of business executives and key opinion leaders. Some of these faces you may recognize. So we're addressing central retinal artery occlusion. This is an ophthalmologic emergency and an important cause of acquired irreversible blindness. 90% of occlusions resolve spontaneously within 72 hours. But at that point, as you know, the damage is already irreversibly done, leading to total or partial blindness. In fact, 80% of patients have final visual outcome of less than count fingers. So there is a window of intervention between 24 to 36 hours. However, this is a statement from American Academy of Ophthalmology. There are no proven therapies or treatments for the ocular manifestations of retinal artery occlusion. So introducing oxygen nanobubbles. This is a therapy that we devised. It's an injectable solution of oxygen encapsulated bubbles for targeted oxygen delivery into the eye. This will be delivered through pars plana directly into the vitreous cavity and will mitigate the damage from the ischemia. So this solution will be stored in physicians' offices, in the ERs, for immediate availability. We devised these particles to be non-toxic. They're made out of dextran shell with oxygen core, and they release oxygen over a prolonged period of time. So CRO is our beachhead market with easiest point of entry as far as regulatory pathway. There are no current efficacious treatments, and this is an orphan condition which provides various advantages through regulatory process. And there is no competition at this point. It's a platform technology applicable to a multitude of other conditions. Even with a beachhead technology as CRO, which is orphan, this is approximately a $200 million a year opportunity. We've been doing research on this for the past 4 to 5 years, and I'll present some of the findings. I won't present the in vitro work, but this is some of the in vivo work. So this is a flat mount of rat retina tissue showing oxygen nanobubbles in gray within the ganglion cells in the retina. So the ganglion cells actually uptake the oxygen nanobubbles. And then on the right here, you see uptake within the ganglion cells and within the microglia as well in yellow. We've used an ischemia reperfusion rat model to show that when injecting oxygen nanobubbles, we're able to oxygenate all the retinal tissues throughout with 2 times improvement in oxygenation. We're also able to show that we're able to preserve function in the rat retina by using electroretinograms. There's also histological evidence of preservation of tissue. On the left, you see the control. In the center, the hypoxic tissues are all disorganized, and on the right, the treated looks pretty much like a control. So at this point, we're at the stage of product testing. The CDMOs are being sourced and currently for scale-up. The ADMET studies are meeting product specifications. We're able to show that there is no oxygen depletion at various storage conditions. The freeze-thaw studies and the mechanical shaking studies look great. Oxygen release studies look good over 6 to 12 hours. Rabbit toxicity studies are normal, so we've done pre-GLP rabbit toxicity studies. This is our timeline. We have our IP secured, and we're expanding. We have our PCT We have proof of concept studies done, publication in a high impact journal. In 2022 now we're engaging FDA. We've submitted an orphan application and waiting for the response. We're doing pre-GLP studies are done. So in 2023 we're going to do all the CGMP work, the GLP work, and by 2024, quarter one, we should have our IND. Future directions will include allied ocular ischemic conditions seen here, which will be a much broader market. And we are looking beyond the eye as well. We have a project that's supported by University of Illinois for wound care. In summary, we're supported by National Science Foundation and angel funding. We're currently raising commercial round of funding. We're first in class. We're addressing serious unmet need with a unique solution. We have a very strong team. All the product specifications are met. The data supports efficacy and lack of toxicity. And there is a clear expedited regulatory pathway. Thank you very much.